Good afternoon. Welcome back to Virology. Today we're going to talk about adaptive immunity. What is adaptive immunity? It is the kind of immunity very different from what we talked about last time. Remember, intrinsic is always present. It's always on in the uninfected cell. And the innate is induced by infection, but it's really rapid. The adaptive is tailored to the pathogen, so it takes a bit more time to be turned on, and it has long-term memory. Now, in recent years, we've learned that innate immunity has some memory, but it's only short-term. Right? So if you get, say, you get infected with SARS-CoV-2 and you recover, you have lingering interferon that will protect you against another infection for like 3 months. And then it goes away. So it's not permanent memory like the adaptive system, which is what we're going to talk about today. Well, permanent, who knows? We'll see. Now, last time I left you with this. We have an epithelial sheet being infected by viruses. The cells sense the infection. They produce cytokines, including interferons, that attract immature dendritic cells. Remember, these are always circulating around everywhere, so they're not too far away. And those immature dendritic cells pick up the cytokines. They have cytokine receptors. They will make interferons themselves to help protect these cells. They also pick up apoptotic bodies or pieces of viruses that are floating around, and that causes them to mature, which we talked briefly about last time. We'll see in a moment. Then they go into the lymph node. So they enter a lymph capillary, they go into a lymph node, which is full of B and T cells, and there they're going to present what they found to T cells. And the T cells will tell them if it's foreign or not. And if it's foreign, then we're going to induce antibodies and cytotoxic T lymphocytes, which are the players for today. So the players, little definitions, leukocytes and lymphocytes. Leukocyte is a general term for white blood cells. Right? We have red blood cells and we have white blood cells. The white blood cells are lymphocytes, neutrophils, eosinophils, basophils, macrophages. They're all shown there. And the lymphocytes are a subset of leukocytes. They're a subset of white blood cells, including T cells, B cells, and NK cells. We talked about those last time. And they all have variable antigen-detecting cell surface receptors. So we're going to talk about that today as well. Okay, those are the players. And how are they made in our body? They're all made from a stem cell in our bone marrow called a multipotential hematopoietic stem cell, which is self-renewing. So it can make more of itself. And it matures to give rise to two lineages. The lymphoid progenitor, which gives rise to lymphocytes, right? NK cells, B cells, and T cells, and eventually plasma cells, which are the antibody-producing factories made from B cells. And then we have the common myeloid progenitor, which gives rise to different cells, including red ones. Look, erythrocytes and macrophages, neutrophils, basophils, and eosinophils. These are the other lymphocytes here as well. So that's where they come from, the bone marrow. And when you get an infection, cytokines go in the bone marrow to turn up this process, to make more of these immune cells. And did you know, as you age, your bone marrow goes away. So if you knock on my legs, it's hollow. It's not quite hollow, but it's more hollow than yours, because as you age, the long bones, the marrow goes away. That's why you have trouble responding to certain infections. Okay, back to innate instruction of adaptive immunity. So remember, we have our dendritic cell, immature dendritic cell. It's picking up signals from infected cells, which we've talked about. That causes it to mature, goes into the lymph node, and now it looks like this. It has the spindly appendages, which is why they're called dendritic cells. It has now it has MHC2 on the surface with a peptide. It's derived from whatever it picked up over at the epithelial sheet. and it will present that peptide to a naive T cell. Naive means it's never seen antigen before. Say the same thing about B cells. Naive B cells have never seen antigen. So the peptide is presented to the naive T cell. If the T cell recognizes it, then the T cell becomes activated and goes out to try and get rid of the infection, because the assumption is that is foreign. And so this MHC class II is very important, right? And normally it's in the endosome, in the immature dendritic cell, and when it gets activated, it goes on the surface so it can present antigen. How it does that, we're going to look at in a moment. Now, something you should know, it's really hard to remember. MHC class I, there are two MHC molecules, which, by the way, are highly polymorphic in humans. Among you, all kinds of different MHC molecules. MHC is on nearly all cells, and 2 is only on these mature dendritic cells, macrophages, and B lymphocytes. So only those cells have MHC2. So how does antigen get presented on MHC molecules? So this is what is called the exogenous antigen presentation pathway. All right, this is a dendritic cell, say, taking up protein by endocytosis. So it's an immature dendritic cell. And that protein is going to be digested eventually into small peptides. And then it's going to be loaded onto MHC2, which you can see here. And that's going to be shipped to the surface to be presented to T cells in the lymph nodes. Okay, so that's where the peptides come from. And some viruses interfere with this process, like cytomegalovirus interferes with MHC2 transcription, so that the cells, that these antigens are not presented to T cells, and so it's basically an immune evasion mechanism. But that's the pathway by which dendritic cells, they're taking up proteins in apoptotic bodies. That's how they get presented on MHC class II. All right, now, these cells, these dendritic cells, and the other sentinels are circulating everywhere, and they're collections of lymphoid tissues in different parts of your body that harbor them and other immune cells as well. So here on the right, for example, is the skin, and we have a special cells. They're basically a form of dendritic cell called keratinocytes that have class I and II. They make cytokines, and they can recognize and present peptides as well. They're also mobile Langerhans dendritic cells. So this is a dendritic cell that is specific to the skin. It will pick up, any proteins and bring them to the lymph node, as you can see there. This is the gut on the left, where there are collections of lymphocytes called Peyer's patches located below the gut epithelium, and these continuously sample antigens that are present in the gut lumen, right? We eat a lot of food, so that has to be sampled for foreign materials and well. And again, these antigen-presenting cells include dendritic cells. They can pick up these antigens and present them to the T cells in the lymph node. And these are called gut-associated lymphoid tissue, or GALT, or mucosa-associated lymphoid tissue, or MALT. Now, when a dendritic cell goes into a lymphocyte and presents antigen to a T cell, and it's recognized as foreign, the T cell multiplies extensively, becomes activated. By the way, when you're, as I said last time, but just to reemphasize, as you are developing, your thymus eliminates all the self-reactive T cells in your body. And so any T cell that recognizes a peptide, the assumption is that it's foreign, because in theory, the self-reactive T cells are supposed to be killed during thymic development. In practice, it doesn't always work properly. As a lot of things in biology, you have T cells that recognize you, and that's where some autoimmune diseases come from. But the idea is that if the dendritic cell finds a T cell that it can react with, then that's foreign. Anyway, when the T cells are activated, they undergo massive proliferation. Within 1 or 2 weeks, you get a 1,000 to 50,000 fold amplification of their numbers. It's huge. And that's why you get lymphadenopathy, swelling of lymph nodes. That's what that means. And I told you the last time, when you get an upper tract infection, these lymph nodes underneath on your neck, they swell. I can feel them right now, but they get much bigger when you have a cold. That's how I can always tell if I have a cold. Now, your body is full of lymph nodes, right? Because they're going to survey, dendritic cells are going to go into them wherever they are in the body and see if they're foreign peptides. So you can see on this illustration, they're all over. But the local lymph nodes are the one that are going to survey the local infection. And most of them you can't feel, right? Most of them are internal. You can feel the ones under your neck, under your chin, under your armpits, in your groin, and that's about it. The ones deep in your intestine, obviously, you can't feel. So if you have gastroenteritis, you can't feel swollen lymph nodes unless you do very unusual procedures, which you're not going to do. Okay, so that's cool. The other thing that's amazing is that, so you have lots of B and T cells circulating in your body all the time, and about 1 in 10,000 or 1 in 100,000 will recognize any given antigen. They will even recognize antigens that are not existing in nature. Like you can make a chemical antigen by synthesis that doesn't exist anywhere, and it will be recognized by some antibody or some T cell within you. It's really, and then that's because of the numbers, which I'll show you in a moment. Really amazing. All right, the first question is, what is a property of innate instruction of adaptive immunity? Presentation of viral peptides on MHC2 to T cells, endocytosis of viral proteins, activation of dendritic cells by cytokines, sensing by toll-like receptors, all of the above. All right, let's have a look at how we did. So the answer is all of the above. All of these are properties of innate instruction of adaptive immunity. Presentation of peptides, but also endocytosis of viral proteins by the dendritic cells, right? Activation of the DCs by cytokines, sensing by toll-like receptors. The DCs have TLRs. They can sense, but also the infected cells have TLRs as well. So they're all part of innate instruction. So I want you to understand that concept, that the innate system via these dendritic cells is instructing the adaptive system on what to do. The dendritic cell has gone into the lymph node. It's presented antigen to a T cell. These are called CD4 T cells. So there are two general types, CD4 and CD8. There are lots of different kinds of CD4s we'll talk about later. But it's the CD4s that receive the peptide from dendritic cells. And so what happens next? That is shown on this side. These are the effectors of the adaptive response. In other words, these are the entities that do things to try and eliminate the infection. So on the right, it's a complicated slide. We'll go through it slowly. So here are all the potential antigens that we have, right, that can be inside of a host, and they're all going to be processed by dendritic cells. And the dendritic cells we've already talked about are bringing the peptide to a CD4 helper cell. So there's a dendritic cell that has done so. And just to emphasize, the naive T cells are produced in the bone marrow, and then they go to the thymus, where they further mature into CD4 and CD8 T cells. These are distinguished by these two cell surface molecules called CD4 and CD8. And you may remember that CD4 is the receptor for HIV, right? And then they go to the lymph nodes. And so when the T helper cell receives a peptide from the dendritic cell, it becomes activated. It makes cytokines of various types. two general classes, Th1 and Th2 cytokines. The Th1 cytokines help the CD8 cells, the precursors, to mature into cytotoxic T lymphocytes, which will go on to kill infected cells. We'll take a look at that in more detail. So Th1 cytokines produced by CD4s allow for the maturation of CTLs. This Th2 cytokines produced by these activated CD4s allow the production of antibodies. And so they help B cells mature into plasma cells, which secrete antibodies, and those antibodies attempt to eliminate the virus or whatever is infecting you. All right, so initially the B cell is present with antibodies in its plasma membrane, and each B cell recognizes one epitope, one specific sequence of amino acids. Now, how are the B cells activated? Those are shown on the left here. So here is a B cell with cell surface receptors. You can see there's some antigen binding to those cell surface receptors. It cross-links the antigen. The antigen is taken up and displayed on MHC2, where it engages CD4 cells. So that's really this part here is shown in some detail. The engagement of the MHC2 and the TCR T cell receptor causes signaling that turns on the synthesis of molecules needed to produce antigen, needed to produce cytokines, helper cytokines like these that mature the B cell to produce antibodies. So the T cells are activated by the MHC presentation by the dendritic cells, and the B cells detect the antigen. They don't necessarily have to be presented by the dendritic cells. So here's an antibody. We want you to be familiar with this because we're going to talk about it a lot. It's a 4-chain molecule, 2 heavy chains and 2 light chains with constant and variable regions. And the variable regions are those that bind the antigen. Hence, they're variable. They are very different from antibody to antibody. And it's divided roughly into 2 parts, an Fc portion, which can bind certain receptors on cells, which we'll see in the coming weeks, and the FAB portion, which binds the antigen. Now, here's a typical antibody response on the left. Say we inoculate a host with antigen A, and then over time we measure the serum antibody titer. And what you will see in 2 to 3 weeks is that the antibody to A will peak. So it takes time. It doesn't happen right away. That's why we have innate responses, because this takes a long time. You'll see why it takes a long time in a moment. These do not remain high. No antibody titer ever remains high. Otherwise, you'd have jelly for blood. So the idea that you can make vaccines that will maintain high levels of antibodies forever is absurd. That's what people are talking about now all the time with COVID. No, it's not happening. The natural function of the immune system, you make antibodies, the levels go down, but you have memory so that you can make them faster next time. And even memory is laggy. It's not 2 weeks, but it's 2 to 3 days. Okay, and then let's say at 7 weeks you inoculate with a mixture of antigen A and a different antigen B. So with antigen A, you get a recall response, a secondary response or a memory response. You can see it's in a couple of days and it's stronger than the primary. And then you get, again, the delayed primary anti-B response. So during COVID, it was all about the waning antibody response, which makes it sound like it's bad. Oh, the vaccine response is waning. What are we going to do? Read an immunology textbook, for one. It's not waning, it's contraction, because you can't keep antibody levels high. We have antibodies against many, many different antigens in us. I'm going to give you a number in a minute. Okay, now I've used some terms So let me define them. The antigen is the molecule that induces the immune response. It can be a protein, can be a DNA, an RNA, a lipid, it could be a polysaccharide. Anything you inject and induces an immune response. The epitope is part of the antigen bound by the antibody or the T cell receptor. It's an epitope. So there on the right, you can see a diagram of an antibody with the antigen combining sites at the tips. And you can see a bunch of amino acids binding to the tips. This is what we call a linear epitopes. The amino acids are all in a row. Sometimes they're conformational epitopes where the amino acids are distant. But typically these antibody epitopes are like 8 to 12 amino acids in length. Okay, a monoclonal antibody is against one epitope. So your serum is a mixture of monoclonal antibodies, many monoclonal antibodies, and we call that polyclonal serum. The serum contains a mixture of monoclonals, but they're all monoclonal in the end. And they have therapeutic value, as you can see. Now, I want to briefly talk about the generation of antibody diversity. So this starts in the bone marrow, where the B cells are maturing. And all the genes to make up the antibodies are at a locus here. That's the germline configuration. And we have like constant regions, variable segments, D and J segments. And these undergo recombination and transcription and splicing to give you the mRNA that is translated to make an antibody. All of this recombination can occur in different ways, right? There are millions of combinations, and plus the splicing can be alternative as well. As a result, in our bone marrow, we make antibodies that would recognize 3 times 10 to the 11th antigens. 3 times 10 to the 11th, okay, by all this recombination and splicing. And that gives us antibodies that are floating around and could recognize anything you throw at them, including, as I said, synthetic antigens. All right, so that's step one. This antibody made by this process is relatively poor. It has low affinity. It's not great, so it has to undergo another step in the lymph node in the germinal center, which is called affinity maturation. And so the idea is that we have these B cells now displaying antigen that have been produced, antibodies that have been produced by this process I've described to you. If the antigen binds with low affinity, it cannot cross-link the antibodies, which are stuck in the cell membrane. So this cell undergoes death by apoptosis. It's eliminated because it's too low affinity. And what happens in these B cells is that the antibody genes are mutated by a specific intentional process involving enzymes that cause mutation. And just randomly, you check all of these new antibodies that are made and pick the highest ones, the highest affinity. So that's called affinity maturation. And when you get high affinity binding, the antibodies are cross-linked, and they can go on and become plasma cells that secrete antigen. So again, you start off with a low affinity antibody, but then you mutate the gene for that antibody. When I mean the gene, it's the last gene encoding this protein after this process. So that's present in one B cell. You mutate it, the protein's made, and it goes through this affinity maturation. And eventually, and this takes time, it can take weeks. You can make really high affinity antibodies that are great for binding to foreign antigens. That happens in the lymph node in the germinal center. Okay, so here are the different kinds of antibodies that we make and the kinetics of their production after immunization. So the first antibody that is made after you immunize, so here we've immunized an animal with poliovirus, and we're measuring the antibody titer days after immunization. And so you see the first antibody to be made is IgM. And that's this one. And that's that first antibody made after VDJ recombination. Low affinity. So what we do is we put 5 of them together to increase the avidity. You got 5 low affinity antibodies, you have high avidity, and it can bind better and be more effective until higher affinity antibodies arise by affinity maturation. So that's the first antibody to be made. And it quickly goes down because it's replaced with IgG, which is higher affinity, and you can see it rises to higher levels. And so here is IgG next to it, the major systemic antibody that is made. We also have other kinds. We have IgE, which we're not going to talk about. It's mainly involved in allergies and parasitic responses. We have IgD, which is the name for the antibody on the surface of the B cell. I just call it the B cell receptor, but it could be called IgD. And then we have IgA, which is present in the mucosa. So it's purposefully sent to the mucosa so that as viruses come in, they can be captured by these antibodies and neutralized. And those are usually dimers held together with a protein called the joining chain. And IgA is a tough one. It's hard to make, and it doesn't last very long. Here's how IgA is made. So a plasma cell has arisen that can make IgA. It secretes IgA just below the mucosal epithelial sheet. These IgA molecules are dimers. They're taken up by endocytosis. They move across to the apical domain where they're secreted, and there they can block viruses if they encounter them. So, what's interesting is that all the respiratory virus vaccines that we have are injected in your deltoid. They're not sprayed in your nose. Well, there's one flu vaccine that's sprayed in your nose, but it's no better than the others. Those vaccines induce IgG mainly, and IgG can also cross these epithelial cells and be present in the mucus, and that's why they work. Otherwise, they wouldn't work because the virus is not getting into the blood. Influenza virus and SARS-CoV-2, respiratory syncytial virus, they're replicating in the mucosa, so the antibodies have to be there to stop them. Now everybody wants to make COVID vaccines that induce secretory IgA. Well, good luck with it, because in most cases the levels go down very quickly and they're not very useful. And people who are deficient in IgA, by the way, they make no IgA. This is like a dirty secret. They don't get any sicker than other people. So tell me what that means, at least for virus infections. All right, what do antibodies do? One thing is they neutralize virus infection. So in the blood or in the mucosa, they can bind to the virus particle and prevent it from infecting, and that can be done by IgG or IgA. And we often say that antibodies are important for reducing the initial infection, not prevent it, because unless you have a very high titer of antibodies in the upper tract, you're not going to prevent it. Infection, you're going to reduce it and therefore influence the subsequent disease. Sometimes antibodies are also important for recovery. Probably together with T cells, they're important for recovery for infection because they sop up all the virus that's being made. Okay, so these are called neutralizing antibodies. And it's the basis for most of the vaccines that we have made. It's how we measure their efficacy. But not all antiviral antibodies are neutralizing. It turns out that some are non-neutralizing, yet they still protect you against disease. I'll give you some examples of that. So what does neutralizing mean, first of all? I'm using this word. I haven't explained it. It means block infection. So here's a plaque assay for poliovirus. On the bottom, we have 4 plates, and we've put enough virus on so we get 11 plaques on each plate. And then above, we have put serum from an animal who's been immunized by poliovirus. And at a dilution of 1 to 10, there are no plaques. So that means the antibodies are neutralizing infectivity. That's what we mean by neutralizing antibodies. They block infection. And then as you dilute out the serum, you can see at 1 to 1,000, you start to get a couple of plaques. And then 1 to 10,000, there's no neutralization. So that gives you an idea of the titers. So when someone says to you, can I do an antibody titer to see if I'm immune to measles? This is what they would do. They would take your blood, take out the serum, do a neutralization assay with infectious measles virus, and calculate the titer that neutralizes 50% of measles. And so if you have 0, you should get immunized for sure. So here's an experiment that shows you how antibodies can protect against disease. Here we have animals that have been inoculated with antibodies and poliovirus. So we're looking at the percent animals paralyzed here and the antibody titer that we have given to these animals. So here's the control set where we give no antibodies, and you can see they're all paralyzed. So that's the control, which shows that your virus is doing the right thing. And then with increasing titers of antibodies, so we're going from from dilute to more concentrated, you can see that the number of animals paralyzed decreases. So at this concentration of antibody, none of the animals are paralyzed. So the antibodies are protecting the animals from paralytic disease. Okay, we used antibodies early in COVID. We did 2 things. First, we got convalescent plasmid. So people who had COVID and they recovered, we took their blood and then we isolated plasma from them. So if you don't know the difference between serum and plasma, serum is what's left after you let the blood clot. You let it clot and you spin out the clot, and that's serum. Plasma is what remains if you prevent clotting and centrifuge out all the cells. So that's what they give people. They would then pool plasma from lots of donors and give it to people to protect them against COVID, getting SARS-CoV-2 infection. Okay, so they were given, this would last about 3 months, and so people were given this prophylactically. So if you had an exposure, you could be given this as well. This fell out of favor, and now it's only used for people with immunodeficiencies. On the right are monoclonal antibodies. We made lots of monoclonal antibodies to different epitopes on the spike of SARS-CoV-2. And these were used early on prophylactically, and they were good. Eventually, all the circulating viruses became resistant to neutralization by these monoclonals, so we stopped using them. Every few months, a new variant would arise that's resistant, and so you can't keep producing monoclonals because the production time is about 6 months. It's very expensive, and then if you use it for 3 months, it simply isn't cost effective. We have better ways to take care of this now. So neutralizing antibodies can work in a number of ways. The most obvious, and perhaps the most common, it seems to be most common for SARS-CoV-2, is that they bind to the virus and block attachment to receptors. So this is using rhinovirus as an example, but it can apply to other viruses as well. So attachment blocking is very common, but you can have other means of neutralizing. You can block endocytosis. You can block uncoating. You can see the antibodies have been taken up with the virus particle in the endosome. You can also aggregate the viruses so they never get into the cell. And some antibodies have been shown to get into the cell and actually neutralize after replication begins. So I just want you to understand there are multiple ways of neutralizing virus infectivity, not just by blocking receptor interaction. However, just listen to how anyone presents, how anyone explains neutralizing antibody. They will all say they block attachment. It's not correct to say that. Right? So where are the epitopes on the viruses to which these antibodies bind? Here on the left is poliovirus, which is a icosahedrally ordered virus. And in white are the epitopes to which antibodies bind. So you can see they're scattered across the capsid, but they're regular, right? Because the proteins that make up the capsid are repeated over and over. On the right are the antigenic sites of influenza virus. This is the HA molecule, the hemagglutinin, which of course is embedded in the virus membrane. And here's the part that would be in the membrane. Then there's a stalk region, and then there is a globular head. And all these colored areas are antigenic sites. That is, they're recognized by antibodies that neutralize infectivity. And for influenza virus, a 1 amino acid change at many of these positions will make the virus resistant to neutralization. And it makes us change. That's why we have to change the vaccine often from year to year. SARS-CoV-2, similar situation. Here's the spike protein of SARS-CoV-2. And these are antigenic, these are actually monoclonal antibodies that were made against different parts of the spike protein. Here's the part embedded in the virus membrane at the bottom. At the top is the part that binds the receptor. Here's the receptor binding domain, RBD. And these are all the different monoclonals in these boxes. And you can see that most of them are directed against the RBM, the receptor binding motif. So that's part of the receptor interaction domain. So that's why I say that most of the antibodies seem to block attachment. And these change too, and that's what makes the monoclonals not usable and perhaps makes us change the vaccine from year to year. Is the structure of poliovirus bound to a monoclonal antibody to show where they bind? So this is the virus particle in the purple is monoclonal antibodies, and 5 are binding around each 5-fold axis of symmetry, as you can see. So each monoclonal recognizes one epitope, and because of the icosahedral nature of the particle, you get repetitive interactions at the 5-fold axis. All right, like everything else in the immune system, viruses evade antibody. How do viruses evade antibodies? Or do they? Sure they do. I've already mentioned that. Here are 3 examples of evasion of antibody by viruses. So on the left is rhinovirus, which exists as over 100 different, let's say, genotypes or serotypes. And each one is not neutralized by antibodies against another. So if you're infected with rhinovirus 1, you recover, you make antibodies against rhino 1, then you're infected with rhino 2 or 100, your antibodies against 1 don't protect you. So you get infected over and over and over. It's a form of immune evasion, just making lots of serotypes. Okay, in the middle is influenza, where I've already shown you this. These are the epitopes in color that are bound by antibodies. 1 amino acid change at any of these epitopes will cause evasion of neutralization. So the mechanism for influenza is antigenic variation, which is caused by mutation of the gene encoding the HA. And that's a random event, and we'll talk about that later. Same thing happens with the SARS-CoV-2 spike. That's shown on the right. And it also undergoes antigenic variation. That's what the variants are that have emerged since the beginning. And all of these red balls, so here the spike is shown as a trimer. So all the red balls are amino acids that change in variants that lead to evasion, and some of these lead to evasion of antibody binding. And so it's why we don't use the monoclonal therapy anymore, and it's why the vaccines have been updated at least a couple times, although I'm not particularly convinced that it was necessary to update the vaccine. I think if you had 3 doses of the original SARS-CoV-2, you're probably okay. It's not clear to me that giving a boost of Omicron is really going to help all that much. We don't have data that says that anyway. All right, so I've talked about neutralizing antibodies, but I don't want you to think that's the only way antibodies work. We're now learning that for many infections, disease can be prevented by non-neutralizing antibodies. I'll give you 3 examples. here's an example of a cell that has Fc receptors on the surface. Now, Fc receptors will bind the Fc domain of the antibody. So you can have antibodies that bind a virus particle, but don't block infection. But those can be bound to Fc receptors, taken up into this cell, which may be a macrophage, and degraded. So that's a mechanism of neutralization. And for some viruses, this has been shown to be the main mechanism for protection against infection. Non-neutralizing antibodies. So that's one example, Fc receptor mediated uptake and degradation of the virus. Another mechanism is called ADCC, antibody-dependent cellular cytotoxicity, again involving non-neutralizing antibodies. So here we have made antibodies against a virus, and we have a virus-infected cell. The antibodies, the virus-infected cell is displaying spike glycoprotein on the surface, which that happens for many envelope viruses, right? The antibodies are binding these, and then the Fc portion of those antibodies are recognized by another cell with an Fc receptor. This is an NK cell, and engagement of these antibodies via Fc receptors leads to killing or apoptosis of this infected cell. The NK cell sends molecules across to the cell that induce apoptosis, so effectively eliminating the virus-infected cell. These can be non-neutralizing antibodies, right? So they won't block infection, but they will lead to lysis of an infected cell. And finally, we have a neat one called netosis, stimulated by IgA. And so what has been observed here is that neutrophils, when they encounter complexes of IgA bound to virus, and these don't have to be neutralizing, again, IgA virus complexes, and it does so via Fc receptors. It shoots out its DNA. So the DNA comes out of the nucleus and makes these nets which trap virus particles. And they've made amazing videos of this. The virus Fc binds and the cells shoot out these, and that's why it's called netosis, right? And it seems to be very effective at minimizing virus loads and for certain viruses. So those are three examples of non-neutralizing activities of antibodies. And the reason I'm emphasizing this to you is because it's becoming increasingly obvious that non-neutralizing mechanisms are important for getting rid of virus-infected cells. Yet, when we test vaccines, the only thing we look at is neutralizing antibody. And in fact, we rarely look at T cells to see how they track with protection against disease. But I think there's much more to a vaccine than making antibodies, and so we really need to change our approach and do other assays that might indicate the value of non-neutralizing antibodies. I know at least one paper recently where a vaccine against Crimean Congo hemorrhagic fever virus, a very severe virus infection in certain parts of the world. The vaccine that is protective in an animal model does not induce neutralizing antibodies. It induces non-neutralizing antibodies, yet it still protects the animals from infection. So I think that's a lesson for us. Okay, next question. Which statement about antiviral antibodies is incorrect? They are important for protection against viral infections. They only neutralize virus infectivity. They may block virus attachment to cells. They can be found at mucosal surfaces. IgM is the first to appear, then IgG. So which one of these is wrong? Okay, let's see how we did. Yes, they only neutralize virus infectivity is wrong, right? So that's why I've been trying to tell you about non-neutralizing mechanisms to block infection. They may block virus attachment, is correct, and IgM is the first to appear, then IgG is also correct. So we've talked about antibodies made by B cells. Now, what about the T cells? That's the other arm of adaptive immunity. So T cells comprises what we call cell-mediated immunity. And we think these are essential for clearing most virus infections, although there's some experiments have shown that they actually can prevent some in the absence of antibodies. And the way it works is that the main cell involved is a cytotoxic T lymphocyte, or the CD8 cell shown here. So CD8 positive T cells are also known as CTLs. They form a synapse with the virus-infected cell, and then they secrete effectors that induce apoptosis of the virus-infected cell. So again, the idea is that you kill the infected cell before it can produce a lot of virus particles. And viruses have ways of getting around this. They have countermeasures, of course. So this depends on the endogenous antigen presentation system, which is affiliated with MHC1, which is present on most cells. So the way this T cell is going to recognize that this cell is infected is that its T cell receptor is going to recognize a foreign peptide presented by MHC1. So MHC1 has to be on most cells because any of your cells could be potentially infected. So how is antigen displayed on a MHC1? So that's called the endogenous antigen presentation pathway. Right? The other one involving MHC2 is the exogenous pathway, where the sentinel cells can pick up proteins and process them inside the cell and present them on MHC2. This is endogenous because it happens in a virus-infected cell. So here we have a virus-infected cell, and there are viral proteins present, right? And some of these are ubiquitinated, which tags them for degradation by the proteasome. They're now made into peptides, which are transported into the ER by a specific transporter protein called TAP1 and TAP2. They're then loaded onto MHC1 and transported through the Golgi to the plasma membrane. And there, the T cell, the CTL, can, via its T cell receptor, recognize that foreign peptide and kill the infected cell. The peptide is presented in the tip of the MHC1, as shown in the upper right here. It's been likened to a hot dog in a hot dog bun. It doesn't look like any hot dog I've ever seen, but okay. The peptide is in blue, and the MHC1, you can see nestling it, is in green. Now, viruses intercept this process in the ways shown by red circles. They can inhibit the proteasome. They can inhibit the TAP that transports peptides into the cell. They can inhibit loading of peptide onto MHC1. They can even inhibit the assembly of MHC1. So lots of ways that particularly the herpes viruses are able to do that. Right, so that's how antigen is presented on the surface of an infected cell so that a CTL can lyse it. So back to the lysis, here's the mechanism. There are two mechanisms by which the CTL lyses the infected cell. So here we have the CTL again, recognizing the MHC1 presented peptide as foreign, and then it's going to induce apoptosis. So one mechanism is by making a lytic pore. So it secretes these perforins, these purple proteins that assemble in the virus-infected cell and make a pore, and basically the contents of the cell leak out, killing it. So that's shown here in A. And then in B, the CTL also secretes granzyme B, shown in red, which goes through the pore and induces apoptosis in the infected cell. So it leads to cleavage of procaspases and the induction of the whole apoptotic pathway. So the CTL releases perforins that build the pore and granzymes that go in and induce apoptosis. So that's how these cells are killed. So how do you counter MHC1? So there are multiple ways. So there's a viral protein. So these are all the viral proteins here. You don't really need to know those. You just need to know that there are viral proteins that will interfere with MHC1 synthesis. As I had told you, they interfere with synthesis of TAP, the transporter, or its function. They interfere with transport of MHC1. They can cause some MHC1 to be retained in the ER. They can move it to the cytoplasm, and they can increase the endocytosis of MHC1, so it's drawn off the plasma membrane into the cell and degraded eventually. So all these rather large DNA viruses and a retrovirus interfere with this so that infected cells are not recognized, right? So that's the key here. An infected cell will not be lysed. So we have two arms of immunity that we've talked about, right? We have antibody and cellular immunity. So which is more important for protecting against disease? And here's an experiment. It depends on the virus, but here's an experiment with Mpox, monkeypox virus, which causes the disease known as Mpox. We vaccinate animals here at day 0 and then do either no immune manipulation or take out the B cells or the CD8 cells. So of course, we're going to prevent antibody production or CTL production. So if you do no immune manipulation, you make really nice titers of neutralizing antibody, and 4 monkeys are not killed by inoculation. So they're all protected from severe disease. If we deplete B cells, we don't make a lot of antibodies, and 3 of the 4 monkeys die. So obviously antibodies are important for preventing severe disease. And then if we deplete the CD8 cells, you get pretty good antibody responses, and all the mice are protected against severe disease. So we conclude that the antibodies here are important for protecting against severe disease. Obviously, you can't do these experiments in humans, right? So we cannot figure out for any virus which is more important. But for some viruses, CTLs are more important than antibodies, and for some, antibodies are more important. So how does the immune system know which one to make? So the answer occurs in the lymph node, where sentinel cells tell the T cells the nature of the invader. So let's take a look at that. Here we have, in the lymph node, certain kinds of TH cells. So I've already told you about CD4 helper cells that interact with the dendritic cells that are presenting antigen, and they stimulate the production of antibodies and T cells. But here we're going to get a little more granular, and we're going to look at different kinds of TH cells. So CD4 is a kind of T helper cell, but there are also many others, as you can see, TH1, 17, 2, Tregs, and so forth. And so depending on the information that's exchanged between the sentinel and the T cell, that will determine whether antibodies or T cell killing is favored, right? So here, for example, here are all the different, here's a naive CD4 cell. It can be induced to make IL-6, which would be valuable for certain kinds of pathogens and tumor immunity. It could be induced to make TH1. Remember, we talked about that before, which helps make CTLs. It can be induced to make IL-4, which would make it a TH2 cytokine for antibody production, and even regulatory T cells to keep everything in check. So it depends on which cytokine is given to the CD4 cell. So that's what these are. I think I said produced, but they're not actually produced. They're given to the naive CD4 cell by the sentinel cell. So there's an example on the right. Here is a sentinel cell. It's also called a professional antigen-presenting cell, macrophage dendritic cell. It is infected by a virus, and in response, or it has taken up a virus, and it's making IL-12 as it presents the antigen to the Th cell in the lymph node. It's going to make IL-12, which causes a Th1 response in the lymph node. And so you don't have activation of a Th2 response, which will give you CD4 cells. So for that virus, the response is skewed to antibody production. And that's how it's done, by the kinds of cytokines that are produced during the interaction of sentinels with the T cells in the lymph node. One more question. For some infections, CTLs are more important for protection than antibody. How is the CTL antibody balance determined? By toll-like receptors, by intrinsic defenses, by autophagy of infected cells, by the mix of peptides and cytokines presented by DCs. It depends on whether the capsid is icosahedral or helical. So what is determining the CTL antibody balance? Okay, how did we do? By the mix of peptides and cytokines presented by DCs, exactly. It's not by intrinsic defenses. That was long ago. And it doesn't matter whether the capsid is icosahedral or helical. So here again, it's at the mix of cytokines and peptides. I don't show you the peptides here, but they're mentioned. That influences the differentiation into Th1 or Th2. And finally, memory. This is the persistence of memory by Salvador Dali. The original does not have viruses in it. So memory is provided by adaptive responses. Long-term memory, as I said, innate responses may have short-term memory, but these have long-term. So that if you are infected with the same virus again later, you have a rapid and specific response. So it's a few days, because you don't have to go through all of that somatic hypermutation to make high-affinity antibodies. They're stored in the form of memory B cells. So you don't have to take 2 weeks to make an antibody. But it takes time, and I'll show you. Memory is the basis for vaccination. You get immunized, you make antibodies and T cells, and those are stored as memory. That's how vaccines work. So this was first realized in an outbreak of measles on the Faroe Islands. The Faroe Islands up here, well north of Scotland, right? So in the 1800s, nobody went there. Maybe one ship a year would go. They were owned by Denmark. It was an outbreak of measles in 1781. Usually someone brought it on one of the ships that came in, because they do need to get supplies now and then. And then for the next 65 years, there were no more outbreaks. Now, there were ships coming over those 65 years, but there were no outbreaks of measles. In 1846, there was another outbreak, but nobody who survived the 1781 epidemic, and there were some, none of them got measles. And so Denmark sent a young epidemiologist to study this, and he said, there must be memory of some kind. Now, we don't even know what viruses are now. We understand what infectious diseases are. We know that in many cases, if you're infected and recover, you don't get it again. So he said, aha, this must be an example of memory, because those people survived, and the other people who didn't have memory, who didn't encounter the virus, or were newborn, were infected. So immune memory can last a long time, as you can see from this experiment, 65 years. And we also understand you don't need re-exposure to virus to maintain memory. Some people used to think that, but you do not. So what is memory functionally? I've already shown you this in another form, but here we have a graph of antibody or T cells. So memory can apply to both. You have your first infection or your first immunization. In 2 weeks or so, you make your first response, antibodies or T cells. It contracts. So you have low levels of antibodies and effector T cells present. And then sometime later, you're infected again. You have a rapid and more robust response, typically a couple of days before you start making antibody from those memory cells. And what does that do? It doesn't prevent infection, but it does make your disease milder because you're going to dampen the amount of virus that is made. It won't be able to spread as well, and you have modified disease. That's the idea. Now, the problem is in elderly people or people with comorbidities, other diseases, memory is not very good. So they can still be overcome by an infection. So you have to take special care for them. But in under 65, the memory should protect you. So what is it physically? So it exists as, for B cells, two forms of cells, memory B cells. So remember, originally we activated some B cells with a specific antigen, with a specific antibody on their surface, those B cells. So we now have clones of these B cells that make the antibody that recognizes whatever virus infected you. And we can store those in the spleen and the lymph nodes, and they will not make antibody unless antigen comes along, unless another virus comes along. They will interact with those cells, and you will get a memory antibody response. Now, you also may remember that these activated B cells became plasma cells, which secrete lots and lots of antibodies. These can also live for a long time, memory plasma cells, and they can be stored in the bone marrow. And again, they will come out when a new infection is sensed. Okay, so that's the B cell component. There's also memory T cells, but I'll talk about those in a moment. I just want to give you an example of the longevity of antibody responses. So here's a table which was made by studying people who had infections, either systemic or mucosal. And they measured antibody, right? And so we're measuring antibody in the serum, and what that's going to do is tell you if you have memory B cells. If you have no memory B cells or no memory plasma cells, you're not going to have any antibody in your serum. Then you have a problem. But for some of these viruses, let's see, polio, 40 years, that's just as long as they went. It could probably go longer. Vaccinia virus, 75 years, smallpox, 40 years, mumps, 12 years, measles, 65 years. That came from the Faroe Island experiment. But then go to mucosal infections, 12 months, 30 months, 3 months, 12 months. It's not really good, right? So there's some difference between mucosal and systemic infections in the ability to induce memory B cells. And we don't know what it is, but it would be nice to find out because we could use it in immunization, obviously. So mucosal infections, the B cell longevity is a problem. We don't know how long COVID vaccine B cell memory B cells are going to last. We know they're there, but not sure how they're going to last. How about T cells? So T cells are a little more complicated. We have a bunch of different kinds of T cells, and this is a graph showing their numbers, the population size on the y-axis with days post-infection. So first infected, you get an expansion of all these T cell types. The orange ones are terminal effector cells. So that would be like this CTL that lyses a virus-infected cell. It does its job, and then it's finished. Eventually, they go away by a month or so. Then we have, these colors are not really great. Huh? I guess this is, this orangey thing must be the resident memory T cells, right? Because the green and the blue are the other two curves. The green and yeah. So this must be the resident memory. These are not circulating, but they're in the organ that was infected. So if you've had influenza, you can find these resident memory T cells, or TRMs, in the lung. So the idea would be, if you get another flu infection, they're going to be reactivated and at the site of infection, which makes great sense, right? And they go down also with time. And then finally, we have effector memory and central memory T cells. That's the blue and the green. So effector memory, TEMs, they circulate in your blood, so they could go potentially anywhere if you got an infection. And central memory are in the lymphoid organs, so they could respond to antigens coming through from infection sites. So the point here again is that the information to, say, kill an infected cell, which resides in the T cell receptor recognizing a viral peptide, that's stored in different kinds of memory T cells. Now, again, we don't know how long memory T cells are going to last for COVID vaccines. They certainly have been measured. T cells have been measured, but it's too soon to know the longevity of them. And these are very important for protecting you against severe disease, because as we'll see later, even though SARS-CoV-2 variants can evade antibody, they do not evade T cells. T cells will recognize any COVID variant, any SARS-CoV-2 variant, and eliminate the infected cell. So very powerful. Okay, now you understand immune responses. So next time we can go on to talk about mechanisms of pathogenesis.